Each codon is 3 bases long. And since each codon is 3 bases long, and there are 4 possible bases, then that means there are 64 possible combinations of 3 bases. 4, 4 times 4 times 4 is equal to 64. So that means there are 64 codons that can exist in the genetic code. Reading them is simplified by the use of this genetic code table. Most amino acids can be specified by more than one codon. If we look at leucine, we see that leucine is in 2 different places on this genetic code table, and each of those places has multiple possible codons. Here we see leucine highlighted or circled. When we read the genetic code table, we read it from the inside out. So we can see that leucine can be coded by 6 different codons. U, U, A. Let me point those out. We see, so we read from the inside out. U, U, A. And then that leads to leucine. We also have U, U, G, which codes for leucine. C, U, C is leucine, as are CUU, CUA, and CUG. All 6 of these codons will code for leucine in a specific position in a protein or in a polypeptide chain. Only one codon, UGG, specifies the amino acid tryptophan. So you can have one codon for one amino acid, or you can have 6 codons, all of which code for one amino acid. Each amino acid is a little bit different. Some of them have multiple codons, some of them have only one codon. However, a specific codon only codes for one amino acid. So anytime you see UUG, it's going to code for leucine. However, leucine can be coded for by 6 different codons: UUA, UUG, CUU, CUC, CUA, and CUG. Although this table applies to most multicellular organisms, including animals and plants, there are slight differences in the code as used by some microorganisms, including prokaryotes, mitochondria, and chloroplasts. Remember, mitochondria and chloroplasts are not individual self-sufficient organisms, but they did evolve, it is thought, from earlier prokaryotes. And so they exist in eukaryotic cells, but they have their own DNA and their own genetic code. If we let's look at a particular codon. Let's look at CAC. So CAC, we see it highlighted in red here. We read from the center to the outside. So we read CAC, and we can see that codon codes for histidine. If we think of the genetic code as a language, a language needs punctuation marks. At the beginning of a sentence, we have a capital letter. At the end of a sentence, we might have a period. In the genetic code, we have start and stop codons that tell when to start making a polypeptide chain and when to stop making that chain. The codon AUG, find the codon AUG. AUG is here. It codes for methionine. Methionine is an amino acid that is the start of most proteins. It is then known as the start codon, AUG. Following the start codon, mRNA is read 3 bases at a time, and amino acids are added one at a time until we reach one of three different stop codons on the mRNA. Any one of those three stop codons can end the translation process. Find the three stop codons. The first one is UAA, the second is UAG, and the third is UGA.